Today I'll be discussing my dissertation research, developing new bioinformatic methods for the analysis of proteomic data, where proteomes are collections of proteins from a sample, and metaproteomes are proteins from a suite of organisms in a complex sample. And I applied these methodologies to Arctic soils in order to understand basic aspects of soil microbiology and how the microbes responsible for the degradation of organic carbon respond to climate change in the Arctic. So first, I will show how rapid Arctic warming has a large effect on plant communities and soil processes in the Arctic. Then I'll briefly summarize current models of soil biogeochemistry and how they can be improved with a greater understanding of microbial processes, which are currently treated as a black box in many of these models. Then I'll explain how a direct study of the proteins can clarify many of these microbial processes, as proteins catalyze the chemical reactions occurring in soils. Next, I will go through the methods that I've developed to facilitate the use of proteomics in complex samples, such as soils. First, I developed software called PostNovo, which improves the accuracy of protein sequence predictions. Then I developed another piece of software called Protein Express to increase the amount of useful data that can be recovered from complex metaproteomes. I applied these to Arctic soil samples that I collected from northern Alaska, and I found key biogeochemical processes and which microbes perform those processes. Arctic warming since the mid-20th century has been quite rapid compared to the rest of the Earth, with an average of about 3 degrees of warming north of the Arctic Circle. Much of this is caused by the loss of Arctic sea ice. As you can see, Antarctica hasn't experienced the same amount of warming. The warmer the land surface in the Arctic, the more vegetation grows there. There's been a marked increase in the height of vegetation, which is relatively low stature normally, throughout the Arctic Circle, with a greater increase in vegetation height with warming. This is also observable from satellite data, where we can see that photosynthetic activity of these communities has increased significantly since warming picked up at the turn of the 1980s. So about 20%. There's been an increase in floral biomass in the Arctic of about 20% since this time, which is relatively small in overall terms of biomass, but as a proportion of the community, obviously quite large. Historical aerial photographs also show this pronounced change, with this photograph from 1966 showing areas of barren tundra, as well as lichen and moss, which is replaced by higher, deeper rooting shrubs for the most part. So not only does the overall biomass increase, but also the composition of the flora changes significantly, with tussock sedges and intertussock mosses and lichens often being replaced by woody shrubs, which can be 1 or 2 meters tall. Much of the turnover in the plant community has involved the replacement of these non-vascular lichens and mosses with vascular deep-rooting plants. Not only does warming have a large effect on the plant communities, but it has the potential to release enormous amounts of organic carbon as greenhouse gases into the atmosphere through accelerated microbial decomposition of soil organic matter. In terms of carbon equivalence, there's about 1.2 times as much organic carbon in permafrost-affected soils as there is in the atmosphere. And here I've shown a soil core that I took to show how most of the organic carbon is concentrated near the surface in the rooting zone. Incubations of soils collected from the Arctic show that most carbon will be lost from the organic layer. A significant proportion can be lost even when incubating at a relatively low temperature of 5 degrees Celsius over a period of about a decade. So over 20% of carbon on average is lost from the organic soils. And incorporating some of this data into terrestrial biogeochemistry models, it's projected that about 9 years worth of current anthropogenic emissions can be lost from the tundra by 2100 and even more further into the future. So this has large implications too for our understanding of warm periods and the distant geological past, such as the Paleocene-Eocene thermal maximum, since there was permafrost at that time as well. And this pool can respond quite rapidly to changes in orbital forcing. So our understanding of the soil organic carbon pool at the most macroscopic level involves the sequestration of carbon dioxide by plants as soil organic matter and the mineralization of that organic matter by microbes largely back into CO2 and other greenhouse gases. The nature of the soil organic matter and its chemical composition has been contentious for quite some time, as it's very difficult to measure, but the current understanding of the soil organic matter indicates that polymers derived from plant and animal residues are degraded by microbes into ever shorter chunks and eventually released as CO2 or incorporated into microbial biomass. And these soil molecules can be sequestered for longer periods of time in aggregates and on mineral surfaces, but there aren't really extremely complex and irresolvable humic molecules that form. It's mostly now thought to be relatively simple and predictable polymers that exist in soils. However, our understanding of the microbes that catalyze this process is relatively poor. So I'd just like to read you a quote from a recent review about soil microbial ecology in Nature. There are numerous individual processes and taxa associated with the metabolism of the thousands of organic compounds found in soil. This complexity makes it very difficult to predict soil function. If, for example, we want to know the fate of labile carbon compounds in soil, which is important in soil carbon models, information about what taxa are present in a given soil sample is unlikely to be useful. So this frames a null hypothesis for my research that the microbial processes involved in organic carbon decomposition are spread relatively evenly across taxa. It's a trait that they can acquire and lose relatively easily, and therefore there is not a strong phylogenetic signal of different chemical transformations. And I will test this hypothesis by using proteomics. It's useful to directly study proteins because they catalyze these chemical reactions in soil. For example, the degradation of cellulose, a major component of the plant cell wall, to simple sugars, glucose. The acquisition of limiting nutrients in soil, so pure phosphate. These are all processes catalyzed by microbial enzymes or proteins. And the traditional method of studying microbial activity in the environment is to look at DNA or RNA, which is really used as a proxy for these reactions, which are encoded by the DNA and RNA through the proteins, which catalyze the reactions. So by using proteomics, we can directly study some of these reactions and maybe even use the presence of proteins as a proxy for many of these different complex reactions occurring in the soil. The general method by which a proteome is acquired and analyzed is that proteins are first extracted from a sample, such as a soil core, and the proteins are digested by an enzyme called a protease into predictable smaller fragments or peptide sequences. So, of course, each protein is a sequence of amino acids. These peptide sequences are then injected onto a tandem mass spectrometer. The tandem part of mass spectrometry refers to the measurement of multiple masses here. So first, the mass of the whole peptide is measured, which gives a mass fingerprint of the peptide. And since each amino acid is associated with a different mass, that can indicate which amino acids are in the peptide. However, the sequence of amino acids still isn't known. Therefore, the peptide is fragmented into even smaller peptides, and the fragmentation spectrum corresponding to the smaller fragments is measured. The amino acid sequence can be reconstructed from the fragmentation spectrum. And my post-novo software facilitates that endeavor. Finally, the whole point of the study is to understand which proteins are present, so these small peptide fragments have to be related back to the proteins from which they derive. And my Protein Express software is involved in that process in my metaproteomic study. So to explain the advances in post-novo, I first have to explain what the traditional method of sequence identification for peptides is. For simple samples, such as biomedical samples, like pure culture of E. coli or human tissues, one knows which proteins are expected to be found in the sample. So one can create a database of the protein sequences expected in the sample, and using the predictable protease cleavage pattern that you use in your own experiment, the individual peptides present in the database can be predicted. Again, each one of these peptides is associated with a unique mass, and the computer can predict a fragmentation spectrum to determine the sequence. These theoretical spectra are then compared to the actual spectra measured from one sample, which is first limited by spectra with overall peptide fingerprints, mass fingerprints, equivalent to the theoretical peptide under consideration. And then the fragmentation spectra can be aligned with strongly overlapping peaks indicating a likely peptide sequence match. So here we find that 2 of the experimental spectra correspond to that specific amino acid sequence. The main challenge of database search, especially for complex samples, is that if you don't know what to expect in your samples, then you have a problem constructing a well-defined database to compare it to. This can be as simple as one amino acid mutation. So even a homologous amino acid mutation produces a large change in the mass, and therefore the peptides will never be linked. This is especially problematic if you've got, say, thousands or even millions of proteins in a sample like a soil, and thousands of organisms expressing those proteins. The alternative, more challenging approach to database search for identification of peptide sequences is the direct sequencing of peptides from the fragmentation spectra without a reference database. You're going from a small set of possible sequences to an extremely large number of possible sequences corresponding to the different fragment peaks. So there are algorithms for de novo sequence prediction, but they don't yield a very large number of accurate sequences, and it's difficult to distinguish accurate and inaccurate sequences using existing algorithms. So I developed a post-processing algorithm called post-novo, which takes the input from multiple de novo sequencing algorithms using multiple theoretical frameworks and machine learning tools. It derives new metrics from the de novo sequences, and it compares them to produce consensus sequences, and those are critical components of the success of the post-novo model. The post-novo score is roughly equivalent to the probability of a sequence with that score being correct. So we can see that inaccurate de novo sequences are assigned low scores, and accurate de novo sequences from these different de novo sequencing algorithms are assigned high scores. The post-novo model does a good job of distinguishing accurate from inaccurate de novo sequences. To validate the model, I used multiple simple data sets for which database search, the traditional method, works very well. That's the gold standard for simple samples like pure cultures of E. coli. That yields peptides in that red star. So that's like the optimal position on these plots. And one can compare the result of the de novo sequencing algorithms in post novo to the optimal yield of peptides. So the closer the curve is to the red star, the better the algorithm performs in these different binary classification statistic plots. And as we can see, post novo outperforms the state-of-the-art individual de novo sequencing tools. Zooming into one of those plots, we can directly see what the increase of the yield of accurate sequences due to the use of post novo. So if one is aiming for a false discovery rate of 20% or precision of 80%, which is, as you can see, quite high for de novo sequencing, then post novo will increase the number of accurate sequences recalled by a factor of 10. So it's a significant improvement on the current use of de novo algorithms. And again, for simple samples, you don't need to use de novo sequencing, but for complex samples where database search breaks down, de novo sequencing might be required. So this raises the question of how we can link these de novo sequences back to the proteins from which they're derived. If one is only dealing with de novo sequencing data in an extremely complex sample, then one might have to resort to comparing the individual peptide sequences to an extremely large database, such as the RefSeq database of basically all known proteins. Now, very short de novo sequences obviously can't be related to unique protein sequences. If you're considering a sequence of length 1, it's going to be found in almost every single protein. And what I found by drawing sequences out of a large database and querying them back against the database is that sequences 11 amino acids long or longer, and de novo sequences really range from about 6 to 20 amino acids in length. These can be uniquely identified across a range of large database sizes. And then I also asked the question, what if the complex sample you're looking at does not have a good representation of proteins in the large database? So if you're looking at a soil, many of those proteins may not be known in the large database, so there's going to be some evolutionary divergence in sequence. And I found that actually the expected level of divergence does not really influence this length threshold that's needed to understand which proteins are related to the de novo peptide sequences. Although I used post novo on my Arctic soils, the most important method I ended up using was a form of database search, and I developed another method called Protein Express to leverage existing metagenomic and metatranscriptomic, or DNA and RNA data from tundra soils in order to form a reference database to identify metaproteomic peptides. So recall that DNA encodes RNA, which encodes protein, so one can predict protein coding sequences from the metagenomes. To understand then which organisms are expressing the proteins, the DNA sequences in the metagenomes must be, quote unquote, binned into organismal categories, or sequences associated with specific organisms. Now, often the sequences aren't long enough, or there just isn't enough information in many of these sequences to assign them a unique organismal identity. So a relatively small fraction of the sequences in the metagenome is binned. So instead of just limiting the size of the database to that small fraction of sequences that's binned or associated with specific organisms, I take all of my protein identifications from all the short and long sequences, a gigantic amount of data, and I similarity search them, or I align these sequences against the sequences in the bins to create a probabilistic measure of how likely a sequence is to be expressed by a specific organism, or how related a sequence is to an organism, to sequences expressed by a specific organism. So in soils, for example, there's a huge strain diversity. So there's a huge diversity of microbes even below the species level, so one might be able to create a bin that doesn't include a large number of the strains. But it's useful to understand that proteins from certain unknown strains are closely related to the species in the bin. Finally, I use existing functional annotation methods to identify biochemical pathways in which the proteins are involved. And I developed a system of functional classification, grouping the pathways into 141 functions of biogeochemical and cellular significance. So 2 of these functional groups are cellulases, or proteins that depolymerize cellulose, and amino acid transporters, or proteins that transport amino acids inside and outside the cell. I collected soil cores from the area of Toolik Field Station, which is the major ecological research station on the north slope of Alaska, north of the Brooks Range. Here's an example of the push core that I used and the sorts of soil cores that I extracted from the ground. I sampled these 18 metaproteomes from 2 sites about 4 miles distant from each other. And I sampled from each of the 3 floral types that I explained before, with the low biomass intertussock vegetation largely comprising moss and lichen, and the higher biomass vascular vegetation types. So I sampled these different vegetation types as a proxy for the changes that are occurring in the Arctic. Again, I searched these metaproteomic data sets against 20 metagenomes and 8 metatranscriptomes, many of which were collected in another study from one of the areas that I sampled, and others were collected about 200 kilometers distant near Fairbanks. And the relative homogeneity of Arctic soils compared to warmer soils greatly facilitated the use of this pooled reference data set for my own samples. I calculate the relative abundances of proteins or protein functions in these data sets by simply summing up the number of spectra associated with the protein and dividing by the total number of spectra in the data set, the common metric that's used. So I'd like to quickly explain what's currently known about microbial diversity in soils around Toolik. So I can first understand the microbial diversity from the metagenomic data sets that I used as a reference. So I can bin sequences related to specific organisms. And I compared this data to the existing 16S rDNA data from other studies. So this is the class level diversity of bacteria from Toolik area samples. And the 16S rDNA gene is a marker for specific taxa. And sure enough, in the metagenomes, I was able to identify most of the main class level groups that are also found in the 16S rDNA samples. So my reference database has 13 bins associated with the main bacterial groups found in Toolik area soils. So first I'd like to show what happens to the overall profile of protein expression in these samples as the flora changes. So based only on the relative abundances of proteins in these 18 samples, the samples sort into low and high biomass floras, which is an interesting result. So these, again, are microbial proteins, and just the distribution of those proteins separates along this main discriminant function into the low biomass samples and the higher biomass samples. Some of the main functions associated with the main discriminant include sugar transporters and EPS production. And actually explain that in a little more detail. So EPS means extracellular polymeric substances. It's the slime that's bound around many bacterial cells, and succinoglycan is a specific sugar involved in EPS around many cells. And one hypothesis for the increasing abundance of these proteins with increasing floral biomass is that microbes are more dependent on interactions with plants. Plants exude compounds into the soil environment, often to create mutualistic interactions with beneficial microbes. And the microbes develop these interactions themselves, triggering plant hormones through a number of complex processes. And EPS production is also involved in many interactions between microbes and plants. So I hypothesize that the increasing abundance of these functions is due to the increasing importance of those interactions. And we can look at some micrographs of where bacteria might live. So they might live on inorganic aggregates or mineral grains, or they are often found in association, in close physical proximity, at least, with a root. So to understand which taxa are responsible for changes in protein expression, I developed a method called taxonomic fidelity, which is the overall abundance of the function from the spectral counts simply multiplied by the relatedness of the identified protein sequences to those taxonomic bins, which are a subset of the metagenomic data. And that metric is an alignment score, or a similarity score between those sequences. So I use fidelity as a measure of protein expression by different taxa. So what I do is I will show the results for comparing these fidelity scores normalized in two different ways between the different floras. So when fidelity scores for a function are normalized across taxa, they indicate the relative expression of each function by different taxa. So if one is considering the sugar transporter functions and normalizing across taxa, we actually see in the data that the rhizobiales, a class of bacteria, class of proteobacteria, are the predominant producers of sugar transports in both floras, and other bacterial groups are relatively minor contributors to the production of sugar transporters. If you normalize in a different way, so normalize across functions, then you find the relative expression by each taxon of the different functions. So now we're just considering the rhizobiales. And even though the rhizobiales dominate the expression of sugar transporters, since the overall abundance of the transporters increases, the relative importance of the sugar transporters increases from low to high floral biomass. So not only considering just one protein or one taxon, but considering the suite of all proteins or all taxa, we can produce a multivariate decomposition of the data, which shows that considering protein expression within individual organisms, so relative protein expression within the individual organisms, the floras separate nicely, whereas considering the distribution of expression of a function across different taxa, they don't separate well. And again, this is because certain functions are expressed by certain microorganisms in general. The bacteria appear to be found in all of the different environments, but they have phenotypic plasticity. They upregulate and downregulate the expression of proteins that are involved in specific chemical reactions that they dominate in the different environments. Okay, so I'm going to continue using this fidelity data. And this is the sort of plot that I'll be showing for much of the rest of the time. So the heat map shows the fidelity data normalized for each function across taxa. So there's one taxon which has the maximum expression of the function on average across the samples. For outer membrane porins, Rhizobiales has the highest expression of those proteins, and the others have lower expression. Then the bars at the bottom show the overall abundance of that function, disregarding taxonomic affinity in the samples. So outer membrane porins, out of these cell envelope related proteins, are one of the most abundant functions. And we can look at one of those functions that increases significantly between low and high floral biomass, the slime production proteins. And as I was asserting earlier, the Rhizobiales dominate the production of those proteins which change from low to high biomass, and the other groups have relatively minimal expression of that function. To understand the validity of the sequence similarity score used in constructing the fidelity metric, we can look at certain functions that are expected to be expressed by certain organisms. Gram-positive bacteria only have one cell membrane. They are not expected to produce proteins associated with construction of the outer membrane. And indeed, looking at the red boxes, the actinobacteria, the gram-positives in the group, have relatively low expression of outer membrane-associated proteins. The bacteroidetes and Myxococcales move around in a very specific way using specific types of proteins. And indeed, those proteins do have strong similarity scores to those specific groups. So that's good. Now, I performed k-means clustering to understand natural patterns in the protein expression profiles, which functions are expressed in a similar way by which organisms. And 3 clusters emerge. Cluster 1 indicates high expression by the proteobacteria towards the upper left of these plots, relatively low expression by non-proteobacteria. Cluster 2 is indicative of relatively even expression between the groups. And cluster 3 is indicative of relatively low proteobacterial expression near the top and a higher non-proteobacterial expression. So in terms of core cellular functions, most of the main functions have the highest expression levels in the acidobacteria, but relatively high expression levels in the proteobacteria as well. Considering carbon metabolism proteins, the relative number of functions associated with either the proteobacteria or the non-proteobacteria increases, and I'll get into this in a bit. The same for nutrient metabolism, where functions highly skewed towards the proteobacteria predominate. So considering core cellular functions, so transcription of DNA to RNA, translation to proteins through the ribosome, we find that the acidobacteria are the most active group overall, which does not exactly mesh with the results from the 16SR DNA data of bacterial diversity, which also are a proxy for abundance. From that data, one would expect a more even distribution of activity between these different groups, whereas we find that the acidobacteria, looking at these key cellular functions, have the highest activity. So the ribosomal proteins, the most abundant function I find in my data sets. Indeed, they're highest in the acidobacteria, the same with RNA polymerase, other key abundant functions. Returning to the null hypothesis that the different taxa are expressing proteins basically in relation to their overall cellular activity regarding the degradation of soil organic matter, so your carbon metabolism. We can test this hypothesis using the data. Another way of saying it is that there's no resource partitioning between the different microbial groups. So what I did here is I simply subtracted the columns from the previous plot, or from those columns in the previous plot, I subtracted the ribosomal column, where the ribosome, again, is a proxy for overall cellular activity. So in the case of many of these functions to the left, we see relatively higher protein expression by the proteobacteria, relatively lower expression by non-proteobacteria. And when the non-proteobacteria dominate a function even more, the dominant function will stay 1, so it'll stay white, but the relative expression of the function by the other groups decreases, so it's blue or negative on the scale. And we see that indeed there are certain functions that are partitioned more to the proteobacteria and more to the non-proteobacteria relative to overall cellular activity. So there's some niche partitioning of key organic matter degradation functions in these soils. To understand what the proteobacteria are doing, first look at the expression of proteins involved in the degradation of small organic compounds, 3 carbons or less, such as acetate, methanol, and carbon monoxide. So carboxytrophy, or the catabolism of carbon monoxide, is dominated by the rhizobiales. Methylotrophy, the degradation of methanol by beta proteobacteria and other proteobacteria. And so we see for all of these blue bars, except one which is relatively low in abundance, they are located, the catabolism of these small molecules is concentrated in the proteobacteria. Likewise, the transport, this is interesting, the transport of simple sugars, especially ribose, shown here, is dominated by the proteobacteria. So again, they're involved in the consumption of small soluble molecules. However, they're not involved in the production of these simple sugars from polysaccharides. That is concentrated in the non-proteobacteria, specifically the acidobacteria. So there are major groups of polymers found within plant cell walls that comprise a large proportion of soil organic matter, these being cellulose, hemicellulose, pectin, and lignin. We find that the actinobacteria seem to have the highest level of cellulose debranching, so cellulases, which is essential for the degradation of recalcitrant crystalline cellulose. They also have the highest level of the actual catabolism of simple sugars, interestingly. And beta-glucosidase can be involved in both cellulose degradation and the degradation of those other polymers, but it's relatively high in the actinobacteria. The acidobacteria, on the other hand, degrade more labile polymers. So hemicellulose is shown here, which have a variety of different sugars besides glucose in them, and starch and glycogen, which are more labile monoglucose polymers. So we can see that down here. Many of these functions are concentrated in the acidobacteria. Lastly, the degradation of lignin appears to be dominated by the Burkholderia group of beta proteobacteria and the actinobacteria. Secondly, we want to understand what's happening with nitrogen, which is a key limiting nutrient in tundra soils. So we've just understood how organic matter is being partitioned between microbial taxa. Now we want to understand how nitrogen is partitioned between the taxa. And we can pose the same hypothesis, that there's no resource partition between the groups. Again, we find that this isn't true, as you saw in the initial clusters as well. The proteobacteria have a disproportionate representation of many of these functions involved in the acquisition and use of nitrogen and other non-carbon molecules in the soil. So first, let's consider proteins that are involved in the intracellular use of nitrogen. For example, the protein glutamine synthetase is essential for the production of amino acids and other nitrogenous compounds, and it's the main protein in this major bar for ammonia metabolism. And that protein is distributed relatively evenly across microbial taxa. It isn't strongly partitioned, and it has its highest abundance in a group of acidobacteria. Some of these other key intracellular processes are likewise relatively evenly expressed. Similarly, looking at phosphate and polyphosphate assimilation, so uptake and metabolism, is spread relatively evenly across the different groups. So phosphate is not strongly partitioned. But interestingly, the transport of nitrogen appears to be, or the expression of nitrogen transporters appears to be strongly partitioned, with proteobacteria dominating some of these key functions, such as amino acid transport, which is one of the most abundant functions. And my hypothesis for explaining this, in line with the proposed dependence of proteobacteria on plant exudates and their strong association with plants, is that there's a depletion of nitrogen around roots, which is well documented, especially by late summer when I sampled. Nitrogen in the soil around roots has been mainly taken up by a mixture by plants as well as microbes. And so to acquire this essential nutrient for essential cellular functions, the proteobacteria living in association with the plant roots must have a higher expression of nitrogen transporters to acquire the necessary nitrogen. So I summarized that the plants are diminishing nitrogen around them, and the proteobacteria are living in association with the plant roots. They appear to form biofilms, especially as the plants grow in which become more important in soil microbial ecology as the plants increase in biomass, as vascular plants replace non-vascular plants. They're largely dependent on simple sugars, but they're not involved in the degradation of the complex polymers, which is mainly concentrated in the non-proteobacteria, including the acidobacteria. And so ultimately arriving at the monomers used in soil carbon cycling, the sources of the monomers are from the plant roots and from the degradation of these polymers. So as I showed earlier, taxonomic niche partitioning is relatively invariant among the floras. So the same taxa are present in the different floras, and they have roughly the same suite of functions that they specialize in, and they vary the regulation of the expression of those proteins depending on which environment they happen to be in. So one can imagine that when the abundance of plants increases, the functions associated with the acquisition of plant exudates also increases, as I observed. The proteobacteria focus on the consumption of small solutes, and the rhizobiales in particular are associated with the functions that increase significantly with greening, or the increase in plant biomass. I am hypothesizing that proteobacteria have a large expression of nitrogen transporters due to their competition with plants for scarce nitrogen in the rhizosphere. The actinobacteria and Burkholdeриaceae have some members that participate in plant interactions, the uptake of oxidase, the consumption of other small molecules in the rhizosphere, but they also have members involved in the degradation of cellulose and lignin, which are the most recalcitrant plant polymers in the soil. And finally, the acidobacteria are the most active group overall, which was somewhat unexpected, and they're involved in the degradation of these more labile plant-derived polymers. And this is interesting because the acidobacteria are relatively poorly documented in pure culture, even though they comprise on average about 20% of bacterial sequence reads from soils around the globe. So it's important to understand what this group is doing to understand microbial soil microbial ecology. Okay. So in conclusion, I developed software which increases the accuracy of de novo sequencing, which is an alternate method of peptide sequence identification. I also developed software which was essential for the analysis of complex soil metaproteomes, which leverages reference databases of pooled metagenome and metatranscriptome samples. The taxonomic fidelity metric which I developed is useful for understanding which taxa express which functions and should be further validated with some controlled experiments, but so far it seems promising. Metaproteomics should now be pursued further in Arctic soils to understand whether some of these patterns hold across the Arctic, as well as other soils other soils in warmer climates to understand whether the same patterns of taxonomic niche partitioning are also found in other soils. One can ask the questions of how does activity change seasonally in Arctic soils, and also with secular warming. It would be very informative to look at soils affected by greenhouse experiments that have been erected over the last 3 decades in the Tula area. And that is it. So I'd like to thank the people who have been essential for this research. So first, my advisor, Jake Waldbauer, and my committee, Maureen Coleman, Michael Foote, and David Archer. The Waldbauer and Coleman lab groups for all of their advice and support. Adriana Rizzo, an undergraduate who was involved in some of the post-novo research. Li-Chun Zhang, who is a researcher in the Waldbauer lab, and the funding sources for the lab, and other members of the department who have been integral to my journey here. Jerry Olag, my former advisor Albert, former graduate student Mark, and my family, my wife Alyssa, my parents, and my in-laws. Thank you.
Any questions from the audience?
I have to ask, are those mosquitoes?
You asked the right question, Sue. Yeah, so that's, I think in June. So I intentionally found the photograph with the most mosquitoes in it. But Tuluk has the world record for the number of mosquitoes killed with one swat of the hand. About 250. Because all the water is pooling at the surface on top of the permafrost. So it's sort of a lake up there. Yep.
Maybe this was a question that you were posing in your conclusions, but when you're saying that you're trying to investigate these different vegetation types as a proxy for the changes that are happening in the Arctic as climate warms, I'm wondering if you had some thoughts about how appropriate that is if you're discounting larger scale ecological changes, things like that. Right, so it's a small spatial scale that you're sampling the vascular plant versus little like mosses, whatever.
Yeah.
Is that scale of change expected to reproduce over the large spatial ecosystem scale?
Clearly, there are a lot of covariates there, like water and other things which influence which plants grow where. So it's always going to be a messy approximation of this, the space for time substitutions that people use, and all sorts of ecological research are always going to be inexact in one way or another. But they hopefully provide some insight. And a lot of the changes you see on a small spatial scale are very similar to the ones you see occurring over huge distances in the Arctic.
Are there any faunal changes that you might expect?
So just the general number of soil invertebrates is a lot lower in the Arctic, and animal biomass is way lower. When I was up there for 2 weeks, I only saw a few pheasant things and not even a grizzly bear. And, there are herds of caribou, which are the main food source for mosquitoes. But it's hard to understand how that many mosquitoes can be supported. So faunal changes are relatively insignificant. Like many cold environments on Earth, it's the simple organisms that dominate, the relatively basic and primitive things that are most abundant. Yes?
Do you have a feeling for spatial heterogeneity and the basic patterns you're finding? For that matter, large scale patterns. If you went into this exact same study in Siberia,
Right.
do you think you'd find, is there any evidence from the available literature that suggests you find pretty much the same relationships?
So the most recent functional research that's been done was an enormous amount of data that was collected from a mire in Sweden. And they did a transect from drier to wetter areas. They used tons of metagenomes and metatranscriptomes, which give you a better approximation of microbial activity. And they had some metaproteomes. They weren't using my methods, so the number of actual protein identifications was much lower. However, what they suggest from that data is that the acidobacteria are involved in polymer degradation. They identified many of the pathways of the acidobacteria relative to other bacteria. And there they found that they express xylonases involved in hemicellulose degradation. They also express cellulases, which here seems to be dominated by the actinobacteria. And so there could be changes in niche partitioning, but there isn't a ton of functional data on microbial communities yet because a lot of these methods are still pretty new. The next step in this research I want to do is actually look at the metagenomes and just get sort of crude estimates of relative abundances of potential pathways. When people have looked at a lot of these microbes in the past, they found that a lot of them have many different pathways because soil genomes are relatively large compared to, say, marine genomes. So they have a lot of redundancy and they can do a lot of things because their environment changes a lot. So, of course, the advantage of metaproteomes is it tells you what things are doing at any one time. So you can see how protein expression, how the functionality of the communities changes with time and environments in a way that you can't see with genomes. Yes?
The taxonomic grouping that you were able to distinguish is relatively coarse, and I think a lot of literature has shown that microdiversity is really important among communities.
I know you published on that.
Many other people. But I'm curious how you think your signals might change if you were able to resolve at a higher level, like species or strain, rather than the class levels that you presented, for either taxonomy or functional correlation.
I think it's likely, so for example, in pure cultures, certain strains, even among the same species as you're saying, can do very different things. And of course, the purpose of this was to understand broader patterns, which is why, the problem with soil metagenomes is you really can't, it's really hard to drive out individual genomes because it's very hard to assemble the genomes due to all the strain complexity. So, yeah, I don't know. I don't really have a priori expectations of what it would be like at the strain level. It might be spread evenly across the class, or it might be focused in, say, a single species of the class. It's hard to tell.
Any other questions or so? If not, let's thank Sam for the presentation.